Welcome to part 4 of our 11-part series on intracellular structures, prepared for the European Society of Human Reproduction and Embryology Clinical Embryologist certification. This session is dedicated entirely to the cytoplasm, a compartment so familiar in name that it can be easily underestimated, yet so extraordinary in function that every outcome we care about in the assisted reproduction technology laboratory depends upon it. In the sessions ahead, we will move from molecular architecture all the way through to clinical decision-making. And by the end of this session, you will see the oocyte cytoplasm not as a passive background fill, but as the most sophisticated developmental machine in all of biology. So let's begin. Think of the oocyte as a self-contained city, and the cytoplasm as its entire urban infrastructure. The roads, the power grid, the warehouses, the factories, and the communication networks all packed into a sphere roughly 120 micrometers across. Every major developmental event that carries an embryo from fertilization to blastulation depends on what is already loaded into that cytoplasm at the moment of retrieval. This is the central insight of this session. Cytoplasmic quality is one of the strongest determinants of developmental competence that we have. And unlike nuclear maturation, which we can assess fairly directly by looking for the first polar body, cytoplasmic maturation is largely invisible to us with conventional light microscopy. We catch only glimpses of it through the dysmorphisms we observe, the granularities, the vacuoles, the refractal bodies, each of which tells a story about what is happening or failing to happen beneath the surface. Here is a point worth pausing on. An oocyte can be meiotically mature, with the first polar body neatly extruded in the perivital space, and yet be cytoplasmically incompetent. It has passed the nuclear exam, but failed the practical. And it is this cytoplasmic competence, not nuclear maturation alone, that determines whether fertilization will succeed, whether oocyte activation will fire correctly, and whether an embryo will progress to blastocyst or arrest at the 4-cell or 8-cell stage in frustrating silence. The cytoplasm also functions as the sole provider of machinery for early embryo development until embryonic genome activation, which occurs around day 3 in the human embryo. Until that point, every protein, every enzyme, every structural component the embryo uses is manufactured from maternally loaded messenger ribonucleic acid already present in the cytoplasm at the time of fertilization. The embryo is, in a very real sense, living off its mother's cytoplasmic inheritance. Understanding this framing makes it immediately clear why clinical embryologists must understand cytoplasmic composition and function at a deep level. Because what we see under the microscope and every protocol decision we make from culture conditions to handling temperature is either protecting or compromising this inheritance. Moving forward, let's take stock of everything we will cover in this session before diving into the detail. This session covers 7 interconnected topics, and they build on one another progressively, so it is worth orienting yourself to the journey ahead. We begin with the anatomy, what precisely the cytoplasm is, how it is defined, and how it is spatially organized within the cell. From there we distinguish between 2 zones within the cytoplasm, the endoplasm and the ectoplasm, which have distinct compositions and distinct functional roles that become particularly important when we are thinking about oocyte cortical events and polar body extrusion. We then examine the 4 major functions of the cytoplasm: structural and mechanical, transport and mixing, metabolic and synthetic, and divisional. Each of these intersects with the embryology we care about clinically. After that, we focus on the cytosol specifically, the soluble phase of the cytoplasm, its physical state, its biochemical composition, and its metabolic roles including glycolysis, protein synthesis, and calcium signaling. From the cytosol, we move to cytoplasmic streaming, also called cyclosis, a phenomenon critical for organelle redistribution in the oocyte and for ensuring balanced cytoplasmic inheritance during cleavage. We then turn to cytoplasmic inclusions, lipid droplets, glycogen granules, secretory granules, and pigment bodies, examining each in the context of oocyte assessment. And we close with the clinical framework, how these concepts translate into interpreting oocyte dysmorphisms under European Society of Human Reproduction and Embryology guidelines, and what the current evidence says about their significance for assisted reproduction technology outcomes. Let's begin with the foundation. What exactly is the cytoplasm? Before we enter the content proper, let's be explicit about what mastery looks like at the end of this session, because these objectives map directly onto the kinds of questions you will encounter in the European Society of Human Reproduction and Embryology written examination and in your daily laboratory practice. You should be able to define the cytoplasm precisely and, critically, distinguish it from the cytosol, a distinction that trips up many candidates because the two terms are often used loosely in everyday conversation, but they are not the same thing. You should be able to describe the structural and functional differences between endoplasm and ectoplasm, understanding why these zones exist and what they each contribute to oocyte biology. You should be able to list and explain the four major cytoplasmic functions in the context of gamete and embryo biology, not as abstract cell biology, but connected to the oocyte, the zygote, and the cleaving embryo. You should be able to explain the composition and metabolic roles of the cytosol, including its role in calcium signaling and cell cycle regulation. You should understand cytoplasmic streaming, what drives it, how it is regulated, and what happens to embryo development when it is disrupted. And finally, you should be able to classify cytoplasmic inclusions, nutritive, secretory, and pigment, and interpret their presence in oocytes within the European Society of Human Reproduction and Embryology assessment framework, including being able to discuss vacuoles, smooth endoplasmic reticulum aggregates, and refractal bodies with clinical nuance. These are concrete, accessible skills. Keep them in mind as we work through the material, and by the end of this session, you should be able to answer each of them with confidence. Let's start with definitions. The cytoplasm is precisely defined as the entire intracellular space, excluding the nucleus. It is bounded on the outside by the plasma membrane, the olemma in the oocyte, and on the inside, where the nucleus is present, by the nuclear envelope. Everything in between is cytoplasm. Now, the cytoplasm is not a single substance. It is a composite of four distinct components. First, the cytosol, the continuous, aqueous, gel-like phase that fills the space between organelles. Second, membrane-bound organelles, the mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and vesicles, each enclosed in a lipid bilayer membrane. Third, non-membrane-bound structures, the ribosomes, centrosomes, cytoskeletal elements, actin filaments, microtubules, and intermediate filaments, and processing bodies known as P-bodies, which regulate messenger ribonucleic acid. Fourth, and very relevant to our clinical work, cytoplasmic inclusions, stored metabolites and secretory granules that are neither organelles nor structural components, but temporary residents of the cytosol reflecting the cell's metabolic state. In the human oocyte, the cytoplasm is the largest compartment by far. The human metaphase II oocyte is approximately 120 micrometers in diameter, roughly 10 times the size of an average somatic cell, and the vast majority of that volume is cytoplasm. This enormous cytoplasm is not simply generous packaging. It exists because the oocyte must contain the entirety of maternal provisions for early embryo development. Every mitochondrion, every messenger ribonucleic acid transcript, every structural protein the embryo will use before its own genome is activated must be packed into this single cell. When we talk about oocyte quality, we are largely talking about the quality of this cytoplasmic store. Without cytoplasm, the cell would simply collapse. Organelles could not be positioned, redistributed during division, or held in the spatial relationships that make their functions possible. The cytoplasm is both the medium and the mechanism of cellular life. With that structural overview in place, let's look more carefully at the two spatial zones within the cytoplasm, because they are not uniform in composition or function. The cytoplasm is precisely defined as the entire intracellular space, excluding the nucleus. It is bounded on the outside by the plasma membrane, the elemma in the oocyte, and on the inside, where the nucleus is present, by the nuclear envelope. Everything in between is cytoplasm. Now, the cytoplasm is not a single substance. It is a composite of four distinct components. First, the cytosol, the continuous, aqueous, gel-like phase that fills the space between organelles. Second, membrane-bound organelles, the mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and vesicles, each enclosed in a lipid bilayer membrane. Third, non-membrane-bound structures, the ribosomes, centrosomes, cytoskeletal elements, actin filaments, microtubules, and intermediate filaments, and processing bodies known as P-bodies, which regulate messenger ribonucleic acid. Fourth, and very relevant to our clinical work, cytoplasmic inclusions, stored metabolites and secretory granules that are neither organelles nor structural components, but temporary residents of the cytosol reflecting the cell's metabolic state. In the human oocyte, the cytoplasm is the largest compartment by far. The human metaphase II oocyte is approximately 120 micrometers in diameter, roughly 10 times the size of an average somatic cell, and the vast majority of that volume is cytoplasm. This enormous cytoplasm is not simply generous packaging. It exists because the oocyte must contain the entirety of maternal provisions for early embryo development. Every mitochondrion, every messenger ribonucleic acid transcript, every structural protein the embryo will use before its own genome is activated must be packed into this single cell. When we talk about oocyte quality, we are largely talking about the quality of this cytoplasmic store. Without cytoplasm, the cell would simply collapse. Organelles could not be positioned, redistributed during division, or held in the spatial relationships that make their functions possible. The cytoplasm is both the medium and the mechanism of cellular life. With that structural overview in place, let's look more carefully at the two spatial zones within the cytoplasm, because they are not uniform in composition or function. The cytoplasm is not homogeneous from center to cortex. It is regionalized into two zones, the endoplasm and the ectoplasm, and this regionalization has direct consequences for how the oocyte functions and how we interpret what we observe morphologically. The endoplasm is the central, interior region of the cytoplasm. It is more fluid in consistency, described as soul-like, and it is organelle-rich. The great majority of membrane-bound organelles, the mitochondria, the endoplasmic reticulum networks, the Golgi apparatus, secretory vesicles, are housed in the endoplasm. Because of this organelle density, the endoplasm is the primary site of metabolic and biosynthetic activity, energy production through mitochondrial oxidative phosphorylation, macromolecule synthesis, lipid processing, and calcium storage. In the oocyte specifically, the endoplasm contains the centrally positioned mitochondrial clusters, the elaborate endoplasmic reticulum networks critical for calcium signaling at fertilization, and the maternal messenger ribonucleic acid stores that will drive all preimplantation development. The ectoplasm, by contrast, is the peripheral zone immediately beneath the plasma membrane. It has a more gel-like, viscous consistency and is relatively depleted of large organelles. Instead, it is enriched in cytoskeletal elements, particularly actin filaments, which give it both structural rigidity and the capacity for dynamic remodeling. In the oocyte, the ectoplasm forms what we call the cortical region. This cortex is the site of some of the most clinically important events in reproductive biology. It is where cortical granules are positioned in a single layer beneath the olemma, ready for their exocytotic release at fertilization. It is where the meiotic spindle migrates in late maturation, positioning itself asymmetrically near the cortex so that polar body extrusion removes the minimum possible cytoplasm from the oocyte. And it is where the mechanical rigidity of the actin network resists deformation during oocyte handling, denudation, and intracytoplasmic sperm injection. The distinction between endoplasm and ectoplasm is not merely academic. When we observe central granularity in an oocyte, a dense granular zone in the center, we are seeing something going wrong in the endoplasmic compartment. When we observe cortical abnormalities, irregular surface morphology, or problems with polar body extrusion, we are looking at pathology in the ectoplasmic zone. This regionalized framework helps us localize and interpret what we see. Now let's examine what the cytoplasm actually does. Its four major functional roles. The cytoplasm performs four broad, interconnected categories of function, and it is worth understanding each in the specific context of gamete and embryo biology rather than as generic cell biology. The first is structural and mechanical. The cytoplasm suspends organelles in a hydrated, gel-like medium, maintaining the internal architecture of the cell. It provides turgor that keeps the cell from collapsing, and through its cytoskeletal elements, it allows controlled repositioning of organelles in response to developmental signals. In the oocyte, the cortical actin network is particularly important. It maintains oocyte shape and provides the resistance that allows the zona pellucida to remain intact during retrieval and manipulation. When we cool oocytes below optimal temperature, we depolymerize this actin network, and the structural consequences can include spindle disorientation and cortical compromise, which is exactly why temperature control during handling is not optional, but fundamental. The second function is transport, mixing, and waste handling. Small molecules can diffuse passively across short distances, but in a cell 120 micrometers across, passive diffusion is simply too slow to ensure that signaling molecules, nutrients, and regulatory proteins reach their destinations in time. The cytoplasm solves this through active transport mechanisms and cytoplasmic streaming, which we will discuss in detail shortly. It also facilitates the dissolving and dispersal of metabolic waste products before they are processed by lysosomes or exported from the cell. The third function is metabolic and synthetic. The cytosol is the site of glycolysis, the entire 10-step pathway, which is critically important in the oocyte because early embryos rely heavily on pyruvate and lactate rather than glucose for their energy, and the cytosolic machinery that processes these substrates underpins that preference. Free ribosomes in the cytosol synthesize proteins destined for the cytoplasm itself, the nucleus, mitochondria, and peroxisomes. In the oocyte, this includes translation of stored maternal messenger ribonucleic acid, which is the engine driving all development before embryonic genome activation. The fourth function is cell division. The cytoplasm provides the environment in which spindle assembly, chromosome movement, and cytokinesis occur. It is not a passive backdrop for these events, but an active participant. Cytoplasmic composition, ionic balance, and cytoskeletal dynamics all directly influence the fidelity of meiotic and mitotic division. And cytoplasmic streaming during these divisions helps ensure that organelles are distributed as evenly as possible between daughter cells, a balance that, when disrupted, produces the blastomere asymmetry that morphokinetic time-lapse systems flag as a marker of reduced developmental competence. These four functions are deeply intertwined, and it is the cytosol, the soluble phase of the cytoplasm, that coordinates them. Let's look at what the cytosol actually is. If you were to take an oocyte, gently disrupt its membrane, and then spin out all the membrane-bound organelles by high-speed centrifugation, what would remain in the supernatant is the cytosol. It is the soluble, semi-fluid phase of the cytoplasm, the continuous aqueous medium in which everything else is suspended. The cytosol is predominantly water, but calling it watery is deeply misleading because it behaves as a viscous, crowded gel. The reason for this is the extraordinary concentration of macromolecules it contains. Enzymes, structural proteins, ribosomes, messenger ribonucleic acids, transfer ribonucleic acids, and signaling proteins are packed into the cytosol at concentrations so high that there is very little free water. This is what cell biologists call macromolecular crowding, and it has profound consequences for how fast reactions occur and how quickly signals propagate. Smaller molecules are also present: glucose, amino acids, nucleotides, fatty acids, and polysaccharides, all serving as substrates for the enzymatic machinery also dissolved in the cytosol. Then there are the inorganic ions, and here we need to be precise because some of these ions are the actual regulators of the most critical developmental events we care about. Sodium, potassium, calcium, magnesium, chloride, and hydrogen ions are all present in the cytosol at concentrations that differ markedly from the extracellular environment, maintained by active transport proteins in the plasma membrane. Calcium is perhaps the most important. At rest, cytosolic calcium concentration is approximately 100 nanomolar, vanishingly small. But during fertilization, that concentration spikes to approximately 1 micromolar, a tenfold increase. And this spike is not a single event, but a series of repetitive calcium oscillations that are absolutely required to activate the oocyte. The calcium is released from the endoplasmic reticulum in waves triggered by a sperm-derived factor, and each oscillation drives a specific downstream event in oocyte activation. If the endoplasmic reticulum calcium stores are inadequate, or if the release machinery is defective, these oscillations fail, and despite sperm entry, the oocyte remains inert. This is the molecular basis of some cases of total fertilization failure that we see clinically. Cytosolic pH is also tightly regulated at approximately 7.2. This matters because shifts in pH directly control the balance of kinase and phosphatase activity that governs cell cycle progression. An oocyte with dysregulated cytosolic pH will have dysregulated cell cycle control, and dysregulated cell cycle control leads to abnormal chromosome segregation and aneuploidy. The crowded, dynamic nature of the cytosol is precisely what makes it an efficient biochemical machine. Enzymes encounter their substrates rapidly, signaling cascades propagate swiftly, and the cell can respond to external and internal signals within seconds. Understanding this dynamism reframes how we think about culture conditions, because every perturbation we introduce, from temperature fluctuation to osmotic stress to light exposure, is a perturbation of this finely balanced milieu. Let's now look more closely at the specific metabolic roles the cytosol performs. The cytosol is not merely a medium in which reactions happen. It is itself a major metabolic engine, performing several critical biochemical functions that are distinct from those of the membrane-bound organelles. The first major role is protein synthesis. Free ribosomes, which are non-membrane-bound complexes of ribosomal ribonucleic acid and protein, float freely in the cytosol and translate messenger ribonucleic acids into proteins destined for the cytoplasm itself, the nucleus, mitochondria, and peroxisomes. In the oocyte, this cytosolic translation machinery takes on exceptional importance because the oocyte has spent years accumulating and carefully silencing a library of maternal messenger ribonucleic acid transcripts. These transcripts are held in storage, translationally repressed, until the appropriate developmental signal releases them for translation. The cytosol is where this translation occurs, and the proteins produced drive every cellular event from fertilization through to embryonic genome activation. The quality of this cytosolic translation apparatus is therefore a direct determinant of developmental competence. The second major role is intermediary metabolism. Glycolysis, the complete 10-step enzymatic conversion of glucose to pyruvate, takes place entirely in the cytosol. This pathway generates adenosine triphosphate and the pyruvate that feeds into mitochondrial oxidative phosphorylation. In oocytes and early embryos, glycolysis and the processing of pyruvate and lactate from surrounding cumulus cells are the primary energy strategies. The pentose phosphate pathway, also cytosol-based, generates the reduced nicotinamide adenine dinucleotide phosphate needed for antioxidant defense, protecting the oocyte from reactive oxygen species, and the ribose sugars needed for nucleotide synthesis and therefore deoxyribonucleic acid replication in the developing embryo. The third role is signal transduction. The second messengers that carry information from the plasma membrane into the cell interior, calcium ions, inositol trisphosphate, cyclic adenosine monophosphate, diacylglycerol, all diffuse through the cytosol to reach their effectors. The great kinase cascades that regulate cell cycle progression, including the mitogen-activated protein kinase pathway and the cyclin-dependent kinase and cyclin complexes that constitute maturation-promoting factor, all operate in the cytosol. Maturation-promoting factor activity is the master switch for meiotic resumption, and its rise and fall in the cytosol directly controls germinal vesicle breakdown, metaphase 1 and 2 arrest, and the completion of meiosis at fertilization. Finally, the cytosol serves as the coordinator between compartments. It integrates signals arriving from the plasma membrane, from mitochondria sensing energy status, and from the nucleus monitoring deoxyribonucleic acid integrity, enabling the cell to mount a coherent, appropriate response. When we think about what makes an oocyte truly competent, it is this coordinative capacity of the cytosol, its ability to receive signals, process information, and orchestrate the right responses at the right time, that ultimately matters most. From this dynamic signaling environment, let's move to a phenomenon that physically distributes all of these cytosolic components throughout the cell, cytoplasmic streaming. Imagine trying to supply a warehouse the size of a football field using only diffusion, molecules simply wandering randomly until they happen to reach their destination. In a small bacterial cell, diffusion is perfectly adequate, but in an oocyte 120 micrometers across, or in the enormous cells of certain plant species, diffusion is simply too slow. Evolution's solution is cytoplasmic streaming, also called cyclosis, the active, directed flow of cytoplasmic contents along the inside of the cell. The mechanism is elegantly simple in principle, though exquisitely regulated in practice. Motor proteins of the myosin family attach to cargo, organelles, vesicles, ribosomes, messenger ribonucleic acid complexes, and walk along actin filament tracks, carrying that cargo in a directed manner. In some cell types, kinesin and dynein motors similarly carry cargo along microtubule tracks. The result is a coordinated, flowing circulation of cytoplasmic contents that achieves in seconds what diffusion would require minutes to accomplish. Cytoplasmic streaming is most prominent in large cells where the need is greatest. It is famously studied in plant cells, where it is visible under standard light microscopy as the chloroplasts and other organelles sweep around the cell periphery in a beautiful procession. But it is equally important in the oocyte, where it operates throughout meiotic maturation and early cleavage. In the oocyte, streaming is regulated by maturation-promoting factor and by the cytoskeletal signaling events that accompany meiotic progression. As the oocyte matures from the germinal vesicle stage through metaphase 1 to metaphase 2, the pattern and intensity of cytoplasmic streaming changes, driving the redistribution of mitochondria, endoplasmic reticulum, maternal messenger ribonucleic acid, and developmental determinants to the regions of the oocyte where they will be needed. During cleavage, cytoplasmic streaming plays a critical role in ensuring that when the zygote divides to form two blastomeres, each daughter cell receives an approximately equal share of organelles, messenger ribonucleic acids, and metabolic substrates. This equitable distribution is what we refer to when we talk about symmetric cleavage, and it is one of the reasons why symmetric blastomere size is considered a morphological predictor of embryo developmental competence. Disruption of cytoplasmic streaming through cytoskeletal toxins, temperature extremes, or abnormal cytoskeletal organization leads to unequal organelle distribution between blastomeres. One blastomere ends up mitochondria-rich and energetically capable, the other depleted and compromised. The asymmetric blastomere sizes and cytoplasmic fragmentation we observe in poor-quality embryos are, in part, the morphological signatures of disrupted streaming. This is not merely academic. It is one more reason why precise temperature control, careful handling, and minimization of cytoskeletal stress throughout our laboratory procedures directly translates to embryo quality. With the dynamic processes of the cytoplasm established, let's turn now to its static residents, the inclusions, which tell their own clinical story. Not everything in the cytoplasm is a permanent resident. Alongside the organelles and cytoskeletal machinery, the cytoplasm also houses a class of structures called inclusions, temporary, non-membrane-bound particles and aggregates that appear and disappear in response to the cell's metabolic needs. Understanding what they are and what they represent is essential for interpreting oocyte morphology. The defining characteristic of inclusions is that they lack a lipid bilayer membrane. This distinguishes them from organelles. They have no permanent structural identity. They form when needed and are mobilized or degraded when no longer required. Their type, number, and size at any given moment are a direct reflection of the cell's metabolic state, its nutrient supply, its hormonal environment, and its current level of stress. Inclusions fall into three major categories. The first is nutritive inclusions, which are energy and metabolic reserves, the cell's pantry. These include lipid droplets and glycogen granules. The second is secretory inclusions, products that have been synthesized and packaged and are now waiting in the cytoplasm for the signal to be released by exocytosis. The third category is pigment granules, stored pigments such as melanin and lipofuscin, which serve structural, photoprotective, or, in the case of lipofuscin, are an unwanted byproduct of oxidative damage. What makes inclusions especially clinically relevant in our field is that they are directly visible in the oocyte under light microscopy on the day of oocyte retrieval and intracytoplasmic sperm injection. The European Society of Human Reproduction and Embryology has developed guidelines for assessing oocyte cytoplasmic morphology that specifically address inclusions as part of the dysmorphism scoring framework. Being able to identify and interpret these inclusions, not just note their presence, but understand what biology they reflect, is a core competency for the clinical embryologist. Let's examine each category in turn, starting with lipid droplets. Lipid droplets are among the most abundant cytoplasmic inclusions in the human oocyte, and they serve as the cell's primary intracellular energy reserve. Their structure is unusual. Unlike other organelles, they are bounded not by a phospholipid bilayer, but by a single phospholipid monolayer, studded with specific proteins called perilipins and lipases, including adipose triglyceride lipase. At their core, they contain neutral lipids, predominantly triacylglycerols and cholesterol esters, densely packed and ready for mobilization. When the oocyte or embryo needs energy, lipases hydrolyze the triacylglycerols to release free fatty acids, which are then transferred to the mitochondria for beta oxidation, the metabolic process that extracts adenosine triphosphate from fatty acid chains. Lipid droplets are also important stores of lipid precursors for membrane biogenesis. As the embryo undergoes repeated cleavage divisions, and generates an increasing number of cells. Each new cell membrane must be assembled, and the lipid reserves in droplets provide the raw material. In the human metaphase 2 oocyte, lipid droplets are abundant and physiologically normal. They are typically scattered throughout the cytoplasm and are particularly concentrated around mitochondria, which reflects their functional relationship. The proximity facilitates rapid fatty acid transfer for oxidation. Under light microscopy, they appear as refractal intracytoplasmic droplets, and their presence in moderate numbers in a normal distribution is entirely expected. The clinical concern arises when lipid droplets are excessive in number, unusually large, or abnormally clustered. This pattern of lipid excess, lipotoxicity, is seen with significantly higher frequency in oocytes from obese patients and from patients with polycystic ovary syndrome. The accumulation of excess lipid in the cytoplasm is not simply an aesthetic issue. Lipotoxicity is associated with impaired oocyte maturation, spindle defects arising from disrupted cytoskeletal dynamics, mitochondrial dysfunction, and ultimately reduced blastocyst formation rates. The lipid overload appears to impair the delicate spatial organization of cytoplasmic components that competent maturation requires. These large or aggregated lipid droplets are a recognized oocyte dysmorphism under the European Society of Human Reproduction and Embryology framework. From one energy storage system, let's move to the other, glycogen. If lipid droplets are the oocyte's long-term energy reserve, glycogen granules are its rapid response glucose buffer. Glycogen is a branched polymer of glucose units, stored as dense granules measuring roughly 10 to 40 nanometers in the cytosol. It is synthesized by the enzyme glycogen synthase when glucose is plentiful and mobilized by glycogen phosphorylase when energy demand rises, releasing glucose units that can immediately enter glycolysis. In the body as a whole, glycogen is most abundant in hepatocytes and skeletal muscle. In our clinical context, it is also present in oocytes, granulosa cells, and early embryos, where it plays a nuanced and important supporting role. The granulosa and cumulus cells that surround the oocyte within the follicle actively metabolize glucose and transfer the products, primarily pyruvate and lactate, to the oocyte through gap junctions. This is not generosity. It reflects the oocyte's metabolic preference. Early embryos rely predominantly on pyruvate and lactate as their primary energy substrates in the period before embryonic genome activation. The oocyte is not well equipped to metabolize glucose directly at early stages, and the surrounding cells perform this conversion on its behalf. As development progresses, specifically as the embryo undergoes compaction at around the 8-cell stage, and then proceeds to blastulation, glucose uptake by the embryo itself increases substantially, and glycogen storage becomes more important as a rapidly mobilizable buffer. During compaction and cavitation, the energy demands of active membrane transport and cytoskeletal reorganization spike sharply, and glycogen granules can release glucose units rapidly to meet this demand when oxidative phosphorylation alone is insufficient. Here is the clinical connection. The diffuse or coarse cytoplasmic granularity that we observe in some oocytes under light microscopy may, in part, reflect abnormal aggregation of glycogen granules or protein aggregates in the cytosol. This granularity is associated with suboptimal developmental outcomes, likely because it reflects underlying cytoplasmic disorganization that impairs the normal spatial distribution of metabolic machinery. From energy reserves, let's move to the third category of inclusions, the secretory products and pigment granules. Secretory inclusions are prepackaged products that have been synthesized, processed through the Golgi apparatus, and packaged into vesicles, awaiting the signal to be released by exocytosis. In classical cell biology, examples include the zymogen granules of pancreatic acinar cells, holding inactive digestive enzymes, and the insulin granules of pancreatic beta cells. In reproductive biology, the most clinically significant secretory inclusions are the cortical granules of the oocyte. Cortical granules are derived from the Golgi apparatus and are positioned in a single, ordered layer immediately beneath the olemma. In the cortical zone of the ectoplasm we discussed earlier, they contain a cocktail of enzymes and glycoproteins, including proteases, ovasticin, and components that modify the zona pellucida. At fertilization, when the fertilizing sperm triggers the calcium wave that sweeps across the oocyte cortex, cortical granules undergo exocytosis en masse, the cortical reaction, releasing their contents into the paravital space. These contents modify the zona pellucida proteins, causing zona hardening and the block to polyspermy that prevents additional sperm from penetrating. It is one of the most elegant and clinically important secretory events in all of biology. When cortical granule migration to the cortex is incomplete during oocyte maturation, or when exocytosis during the cortical reaction fails to occur fully, the zona block to polyspermy is compromised, increasing the risk of polyspermic fertilization and abnormal polyploid embryos. This is one reason why cytoplasmic maturation, including the migration of cortical granules to their correct cortical position, is a prerequisite for competent fertilization. Pigment granules represent a more heterogeneous category. Melanin granules and melanocytes serve the familiar photoprotective function. Of more direct relevance to reproductive biology is lipofuscin, sometimes called age pigment, a non-degradable complex of oxidized lipids and proteins that accumulates in post-mitotic and aged cells as a byproduct of imperfect lysosomal processing. Lipofuscin cannot be broken down or exported, it simply accumulates. In oocytes, the structures visible under light microscopy as refractal bodies, bright, highly refractive inclusions in the cytoplasm, are analogous to lipofuscin-like aggregates. They reflect accumulated oxidative damage to cytoplasmic components, and their presence is associated with reduced developmental competence. Refractal bodies are a recognized cytoplasmic dysmorphism under the European Society of Human Reproduction and Embryology framework. Seeing them in an oocyte should prompt careful consideration, though as we will discuss, they are not grounds for automatic discard. Let's now turn to the full clinical assessment framework for cytoplasmic inclusions and dysmorphisms. In clinical practice, the assessment of oocyte cytoplasmic morphology takes place on the day of denudation, typically at the time of intracytoplasmic sperm injection or in vitro fertilization insemination. At this point, the embryologist observes the oocyte under an inverted microscope and records the presence of any morphological deviations from the ideal, what the European Society of Human Reproduction and Embryology and the alpha scientists in reproductive medicine consensus guidelines call dysmorphisms. Dysmorphisms are classified as either intracytoplasmic, within the cytoplasm, or extracytoplasmic, involving structures outside the cytoplasm proper, such as the zona pellucida, polar body morphology, and paravital space. In this session, we are focused on the intracytoplasmic dysmorphisms. There are 6 key intracytoplasmic dysmorphisms to know. Diffuse granularity describes a fine or coarse granular texture distributed throughout the cytoplasm. This pattern is associated with abnormal mitochondrial clustering and is linked to reduced developmental competence. It is one of the most commonly observed cytoplasmic abnormalities in clinical practice. Central granularity is a more localized variant, characterized by a dense granular zone specifically in the center of the oocyte. This reflects mitochondrial and endoplasmic reticulum aggregation in the central endoplasmic region and is associated with poorer outcomes than peripheral granularity. Refractal bodies are bright, highly refractive inclusions scattered in the cytoplasm, representing the lipofuscin-like oxidative damage aggregates we discussed. Their presence signals reduced developmental potential, particularly when they are numerous or large. Vacuoles are membrane-bound, fluid-filled inclusions that appear as dark, optically empty spaces within the cytoplasm. They arise from endoplasmic reticulum dilation, abnormal autophagosome formation, or osmotic injury, and their clinical significance depends heavily on their number and size. Smooth endoplasmic reticulum aggregates, disc-shaped clusters of smooth endoplasmic reticulum membranes, appear as distinct refractal areas in the cytoplasm and are associated with altered intracellular calcium distribution. The current European Society of Human Reproduction and Embryology guidance holds that smooth endoplasmic reticulum aggregates alone should not be grounds for automatic oocyte discard. Lipid droplets in excess, large or clustered lipid inclusions beyond what is physiologically normal, are associated with lipotoxicity and impaired maturation, as we discussed in the context of obese patients and polycystic ovary syndrome. Knowing these 6 dysmorphisms by name, appearance, and biological significance is a clinical embryologist examination essential. Let's now examine the 2 most clinically debated of these, vacuoles and smooth endoplasmic reticulum aggregates, in greater detail. Of all the intracytoplasmic dysmorphisms we observe, vacuoles and smooth endoplasmic reticulum aggregates generate the most clinical discussion and the most examination questions, because neither has a simple, universal management rule and both require case-by-case judgment. Vacuoles are membrane-bound, fluid-filled inclusions that vary considerably in size and number. They may arise through several mechanisms: dilation of endoplasmic reticulum cisternae under stress, aberrant formation of autophagosomes during cellular quality control processes, or osmotic injury during laboratory handling. Small, solitary vacuoles are generally considered to have minimal clinical impact, and oocytes bearing them are typically used without reservation. Large vacuoles, or multiple vacuoles occupying significant cytoplasmic volume, are a different matter. These are associated with reduced fertilization rates, impaired early embryo development, and higher rates of developmental arrest. When a vacuole is present at the time of intracytoplasmic sperm injection, there is a specific practical concern. The injection needle may inadvertently engage the vacuole during microinjection, incorporating it into the injection site. This can disrupt the cytoplasmic environment at the point of sperm deposition and may contribute to abnormal development. Experienced embryologists will adjust their injection technique to avoid vacuoles where possible, selecting an injection site away from any visible vacuolar inclusions. Smooth endoplasmic reticulum aggregates are morphologically distinct from refractal bodies and must not be confused with them. They appear as disc-shaped, refractal clusters, sometimes described as having a slightly flattened or lobulated appearance, and represent aggregations of smooth endoplasmic reticulum membranes. Their biological significance lies in their association with altered intracellular calcium distribution. The smooth endoplasmic reticulum is the primary calcium store in the oocyte, and when its membranes are abnormally aggregated, the spatial distribution of calcium released during the cortical reaction is expected to be dysregulated, potentially altering the pattern of calcium oscillations at fertilization. Early studies raised significant concern about smooth endoplasmic reticulum aggregates, suggesting associations with abnormal outcomes including, in some reports, abnormal placentation. More recent evidence has been less consistent, and the clinical picture has become more nuanced. The current European Society of Human Reproduction and Embryology guidance takes a balanced position. Smooth endoplasmic reticulum aggregates alone should not automatically disqualify an oocyte from use, particularly in circumstances where oocyte yield is low or the patient is of advanced reproductive age. The decision to use or discard oocytes bearing smooth endoplasmic reticulum aggregates must be individualized, taking into account the full clinical context. How many oocytes are available, what the patient's history is, what alternatives exist, and must be accompanied by appropriate patient counseling about what is known and what remains uncertain. This nuance is not weakness, it is clinical maturity. The evidence base for oocyte dysmorphism management continues to evolve, and the embryologist's role is to interpret morphology in context, not to apply rigid binary rules. As we connect these morphological observations to their biological consequences, let's now look directly at cytoplasmic maturation and why it is as important as the nuclear maturation we can more easily confirm. We can confirm nuclear maturation with confidence. The presence of the first polar body in the paravitallin space, tells us the oocyte has completed germinal vesicle breakdown, progressed through metaphase 1, and is now arrested at metaphase 2, ready for fertilization from a nuclear standpoint. But here is the uncomfortable truth. Nuclear maturation and cytoplasmic maturation, while normally synchronized, can become dissociated. And an oocyte with a first polar body and no apparent morphological abnormalities may still be cytoplasmically immature, silently incompetent beneath an apparently normal surface. This dissociation is more common after ovarian stimulation than in natural cycles. When we administer exogenous gonadotrophins to recruit a cohort of follicles that would otherwise not ovulate, some of those follicles may not have had sufficient time or the optimal hormonal milieu to complete cytoplasmic maturation even as the nucleus completes its divisions. The oocyte looks ready. It is not. What does cytoplasmic maturity actually consist of? There are 5 key markers. First, appropriate mitochondrial distribution. As the oocyte matures, mitochondria redistribute from a pericentral clustering pattern toward a more peripheral dispersal, ensuring that energy production capacity is appropriately distributed throughout the cytoplasm. Second, cortical granule migration. Cortical granules must migrate from their interior positions to form the single-ordered cortical layer beneath the elemma that enables the cortical reaction at fertilization. Third, completion of endoplasmic reticulum maturation and calcium store loading. The endoplasmic reticulum must not only be structurally mature, but must have accumulated the calcium stores that will be released as oscillations at fertilization. Fourth, redistribution of maternal messenger ribonucleic acids and proteins to their appropriate cytoplasmic regions. Developmental determinants must be positioned where they will be needed at the right developmental stage. Fifth, cytoskeletal reorganization. The actin and microtubule networks must remodel to support the asymmetric positioning of the meiotic spindle and the mechanics of polar body extrusion. When one or more of these elements is incomplete, when the oocyte is cytoplasmically immature, the clinical consequences are significant. Fertilization rates are reduced even after intracytoplasmic sperm injection because the problem is not sperm entry but what happens afterwards. Oocyte activation fails or is incomplete because the calcium oscillation pattern is insufficient. Early embryo arrest occurs at higher rates because the cytoplasmic provisions are inadequate for sustained development. Blastocyst formation rates are impaired despite normal fertilization morphology. These patterns are frustrating because they mimic the clinical phenotype of poor quality gametes without any obvious morphological warning sign on the day of intracytoplasmic sperm injection. It is precisely this challenge that has driven research into artificial oocyte activation. Using calcium ionophores, such as a 23187 or ionomycin, to pharmacologically trigger the calcium oscillations that a cytoplasmically immature oocyte cannot generate itself. Artificial oocyte activation is used in selected clinical cases where total or near-total fertilization failure has occurred in previous cycles, and where the pattern of failure suggests oocyte activation deficiency. Understanding cytoplasmic maturation in its full complexity is what allows us to diagnose these situations and counsel our patients appropriately. Let's bring all of this together in the final clinical synthesis. Every step of the in vitro fertilization process, from follicular stimulation to retrieval, denudation, intracytoplasmic sperm injection, embryo culture, and cryopreservation, interacts with the cytoplasm. Understanding the specific mechanisms through which cytoplasmic quality influences outcomes allows us to design protocols that protect rather than compromise the cytoplasmic integrity on which developmental success depends. Mitochondrial sufficiency is foundational. Mitochondria generate the adenosine triphosphate that powers spindle assembly, chromosome movement, and cytokinesis. When mitochondrial density is inadequate, whether due to maternal age, mitochondrial deoxyribonucleic acid deletions, or cytoplasmic abnormalities that disrupt mitochondrial function, spindle assembly fails, cytokinesis is incomplete, and embryos arrest early. Mitochondrial deoxyribonucleic acid copy number in oocytes is currently being explored as a biomarker of developmental competence, though it has not yet entered routine clinical use. Calcium signaling competence is perhaps the most direct link between cytoplasmic quality and fertilization outcome. The volume of endoplasmic reticulum calcium stores and the sensitivity of inositol trisphosphate receptors on the endoplasmic reticulum membrane determine the amplitude and frequency of the calcium oscillations triggered at fertilization. If the stores are undersized or the receptors are insensitive, oscillations are weak or fail to sustain. Insufficient oscillations mean failed oocyte activation. The zygote is never properly switched on. This is why artificial oocyte activation with calcium ionophore is used in cases of repeated total fertilization failure after intracytoplasmic sperm injection, where the pattern points to activation deficiency rather than sperm dysfunction. Cytoskeletal integrity directly affects chromosomal fidelity. Disrupted cortical actin leads to abnormal spindle positioning, which leads to unequal chromosome segregation and aneuploidy. Temperature fluctuation is one of the most potent disruptors of the meiotic spindle. Microtubules depolymerize rapidly when temperature drops even a few degrees below 37 degrees Celsius. This is the biological justification for strict temperature control throughout oocyte handling. A pre-warmed microscope stage, pre-warmed dishes, minimize time outside the incubator, and careful attention to every step in the intracytoplasmic sperm injection procedure. It is not ritual, it is cytoskeletal protection. Finally, the balance between reactive oxygen species and antioxidant defenses in the cytoplasm is a critical determinant of long-term cytoplasmic integrity. Reactive oxygen species damage proteins, lipids, and nucleic acids. The antioxidant enzymes that neutralize them, superoxide dismutase, catalase, and glutathione peroxidase, are cytosol-based. Culturing embryos under reduced oxygen tension, 5% oxygen rather than atmospheric 20% oxygen, significantly reduces cytoplasmic oxidative stress and improves blastocyst formation rates. This is why all contemporary high-quality embryology laboratories use low-oxygen incubation conditions. The rationale goes directly back to cytoplasmic chemistry. Before we close Part 4, let's consolidate what we have covered, because the examination will test your ability to integrate these concepts, not merely recall individual facts. The cytoplasm is the entire intracellular space excluding the nucleus, comprising the cytosol, membrane-bound organelles, non-membrane-bound structures, and inclusions. It is the operational environment for all cellular processes and, in the oocyte, the sole provider of maternal provisions for early embryo development. The cytoplasm is spatially regionalized into the endoplasm, organelle-rich, fluid, and metabolically active, and the ectoplasm, actin-enriched, gel-like cortex housing cortical granules and supporting the asymmetric events of polar body extrusion and the cortical reaction. The cytosol is the metabolically active, crowded aqueous gel that performs glycolysis, free ribosome-based protein synthesis, calcium signaling, and cell cycle regulation. Its ionic composition, particularly calcium concentration and pH, is not incidental background chemistry, but the direct regulator of the most critical events in oocyte maturation, fertilization, and early development. Cytoplasmic streaming distributes organelles and developmental determinants throughout the oocyte and ensures balanced cytoplasmic inheritance during cleavage. Disruption of streaming produces blastomere asymmetry and reduced developmental competence. Cytoplasmic inclusions, lipid droplets, glycogen granules, secretory granules, and pigment bodies reflect the metabolic state of the cell and are directly accessible morphologically. Abnormalities in their number, size, or distribution are the basis of the cytoplasmic dysmorphism framework. The European Society of Human Reproduction and Embryology recognizes 6 key intracytoplasmic dysmorphisms: diffuse granularity, central granularity, refractal bodies, vacuoles, smooth endoplasmic reticulum aggregates, and excess lipid droplets. None of these are grounds for automatic oocyte discard. All require clinical contextualization. Cytoplasmic maturation is a prerequisite for competent fertilization, oocyte activation, and preimplantation development, and it is independent of nuclear maturation. An oocyte at metaphase 2 can be cytoplasmically immature, and this dissociation accounts for some of the most clinically frustrating patterns of fertilization failure and early embryo arrest. Every step of the assisted reproduction technology process introduces cytoplasmic stress. Protocol optimization at each stage, temperature control, low oxygen culture, gentle handling, is fundamentally an exercise in cytoplasmic protection. That brings us to the end of Part 4 of the Intracellular Structures series. The cytoplasm, far from being the unremarkable background of the cell, is revealed as its most complex and clinically consequential compartment, the inherited machinery on which every developmental decision depends. In Part 5, we will shift our focus inward, moving from the cytoplasm to the structure that it surrounds and serves, the nucleus. We will examine its architecture, its organization, and its role in governing the genetic program of the developing embryo. The nucleus is the instruction manual. The cytoplasm, as we have learned today, is the factory that reads it. Both must be intact and functional for development to succeed. Thank you for your attention throughout this session, and I look forward to continuing our exploration of the cell's intracellular world in Part 5. Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.